Good afternoon, everybody, and thank you for coming to the first opening event of Postdoc Appreciation Week. I'm Jason Lewis. I'm the Deputy Director of SKI, and I have the absolute pleasure of overseeing the OSET office, which I know you've all interacted with, and the amazing team with Yahara, Ina, Thaliana, and Sandy. Sandy's a little distracted right now because her daughter-in-law is in labor. So no matter the presentations today, is it your son's first? She's so distracted right now. Okay, yes, she's pushing. Okay, a huge thank you. This is the week to celebrate the postdocs. We have 549 postdocs at MSK, and the number which I got this morning, which amazes me, this represents 57 nations across the globe. So this is a truly international group of workers that we have at MSK, but you're also highly trained investigators. You drive innovation within laboratories, and the MSK mission couldn't be possible without you and your hard work, your innovation, and everything that you do. Today, Yahara is going to introduce the judges, but I'd like to thank them for coming today, some outside judges to give a completely impartial voting on the Postdoc Slam today. Thank you to Dr. Vickers and the leadership team for also coming. I do want to mention that, as I said, we had large numbers of postdocs, and your contributions are amazing, but I know that a lot of people have questions about things like job security, funding situations, and other challenges that are currently hitting our postdoc population. So we will have on October 9th a town hall that's organized with your amazing Postdoc Association. It will be in this room where we're going to address many of these concerns and also present to you the results of the survey that we recently conducted. We want to make sure that we hear your concerns, we can act on them, and we can really do something to enhance the postdoc experience here at MSK. So with that said, it's now time to recognize your efforts. And we're now going to have the Postdoc Slam. Yahara is going to explain how we got to where we are. We're going to highlight these 10 today, as we also have highlighted and will highlight other postdocs through our social media platform over the next few months. I'm going to step aside and let you know that I have no input whatsoever into the decision today. So my own lab are presenting, so I will be completely impartial. I'll go sit at the back like a nervous parent, like we all do, those of us run labs, as they do that. So you won't see me on stage again today, but I do want to remind you that on Thursday, we have another phenomenal presentation from our postdocs. Here are three here with a keynote speaker, Lydia Lynch, who's going to talk about her research. And so thank you very much for coming this afternoon. I'm now, as I said, I'm going to sit back, be a nervous parent, and I'm going to hand over to Yahara to start off the Postdoc Slam. Thank you.
Thank you, Jason, and thank you, everyone, for being here today. Today is a very awesome day for the team. We call it one of our favorite days in the office. Everyone is like hands on deck trying to make this happen. And when we walk into the room today, we are like, this is going to make a special Postdoc Slam because the lights on the auditorium are not working. So it's a candlelight Postdoc Slam. How amazing that is. As Jason said, this event is something that we started in 2018. So now in our 8th year, we have had over 150 people that have presented in the different rounds of the Slam. We have had 8 phenomenal scientists win first place. They all happen to be women, so go women. No pressure on the gentleman presenting today. And then this is what the Postdoc Slam is. It's an event modeled after the 3-minute thesis competition. The presenters are going to have 3 minutes and 1 slide aimed at a non-scientific audience. The majority of us in the audience probably have some kind of touch on science, but this event is more for the people that might not have been at the bench day in and day out. So keep that in mind as you are hearing their presentations. They're going to be judged for their content, their storyline, and their overall delivery. We kind of want those good stories in front of there. And the winner is going to walk out of here with $3,000, the second place with $1,500, third place with $1,000, and the audience in the room is going to be able to vote for the people's choice. So this is basically you tell us the people that you want to raise as a people's choice winner. We have in the room 6 wonderful judge members who are going to be doing a really hard job of selecting who wins, and it's a tough competition out there. I want to publicly thank them all: Sean Decatur, Ellen Futter, Claudia Oberstrom, Alan Snitcher, George Walker, and Peter Weinberg. Thank you so much for donating your time. This is a delightful event, so I hope that you walk out of here not only thinking MSK is amazing, it is, but also knowing that our scientists are top of the line in the work that they do in the lab, and also when we put them in front of amazing audiences as well. So thank you. And with that, I also want to thank all the people that got us here, including the semifinal judges, Shelly Anderson, John Lasky, and Cardi Smith, here from our own team. Then also in the qualifying round, we were able to bring Daniela, who was a winner from last year Postdoc Slam, back into the room to help their peers get here today. And we have another postdoc from Levine Lab, Pablo, helping us with that. Thank you, Daniela. Thank you, Pablo. And we also have a wonderful development team, the design and creative service, the comms team, and the digital team that are working up out there to record the event, help us polish it, make it awesome. So thank you, all of our colleagues. And with that, it's showtime. We have 10 speakers today. I'll introduce them. They'll take the stage. She's back to back. And at the end, as I mentioned, I'll escort the judges out of the room for voting, and the room stays with Ina, who's going to lead the People's Choice. Are we ready? It's my pleasure to introduce our first speaker for the Postdoc Slam today, Megan Elizabeth Kelley from the Sfeir Lab in molecular biology, is talking about losing our edge, combating drug resistance in cancer.
In 2013, actress Angelina Jolie announced that she'd had a double mastectomy after finding out she was a carrier for a BRCA mutation. She did this because carrying a BRCA mutation can increase a person's risk of developing breast cancer up to 80%. Now, a double mastectomy is incredibly effective, though still not 100%, and BRCA mutations can increase your risk of developing other types of cancers, including ovarian and prostate, so we need therapies. The good news is that there are very effective therapies, a class of drugs known as PARP inhibitors, that can be used to treat BRCA mutant cancers. The bad news is that resistance often occurs with this treatment. To understand where this resistance is coming from, we need to look at our genes. Now, our genes are written in code, a code of 3s, where there are 3 bases or letters encoding for a piece of a protein. When a mutation occurs that shifts this code, deleting a piece, we get nonsense happening afterwards, pieces of proteins that don't belong there. This renders BRCA non-functional. Now, this is good news for treating with PARP inhibitors because BRCA is usually involved in DNA damage repair, and PARP inhibitors increase a cell's reliance on DNA damage repair pathways. But sometimes BRCA gets another mutation that shifts the code again, and we get a predominantly functional protein again. This means PARP inhibitors are no longer working, but we still have cancer, so there's a need for additional therapy. This is where my lab comes in. We study a process known as MMEJ. It's a type of DNA repair pathway where 2 distant segments that are identical are joined together with the help of a protein we identified known as POLQ. When this happens, you get a deletion often that's very similar to the types of deletions that we see in BRCA reversions or resistance mutations. So we think that MMEJ might be causing this. Now, the good news is that we have inhibitors for POLQ. We can combine these inhibitors with PARP inhibition, where PARP will kill off the BRCA mutant cancers, and POLQ inhibitors will hopefully prevent MMEJ from occurring, and if that's what's causing these resistance-conferring mutations, we won't get the resistance that comes back. And so what we're doing in our lab is engineering cells that have these BRCA mutations and a fluorescent reporter that tells us when the resistance is occurring. We can sort that out, sequence them, and see if our hypothesis is correct, and MMEJ may be driving this type of resistance. By combining these 2 together, we're hoping this will give us back our edge when it comes to treating BRCA mutant cancers. Thank you.
Our next participant comes from the Kolesnick Lab in molecular pharmacology. Honorine Dushime will present on Lucky is the human whom the Muse saved.
Good afternoon. This year marked the 80th anniversary of the destruction of Hiroshima during the Second World War by a nuclear bomb. Thousands and thousands of people died due to the bomb explosion, but more and more died due to radiation poison from the bomb. So today there is an increasing risk of mass exposition to high dose radiation due to a more nuclear weapon that are more powerful than 80 years ago. There is also terrorism threat, but an increasing risk of radiation accident due to the impact of extreme weather on nuclear facility. So what's happened when people are exposed to high dose radiation? High dose radiation will destroy the intestine, which is one of the most radiosensitive organ of our body. Once the intestine is destroyed, people develop a disease called gastrointestinal syndrome and died within 10 days. And currently there is no effective treatment of gastrointestinal syndrome. So my studies was focused on using cell therapy, and this method consists in giving healthy cells in order to restore normal function of injured organ or tissue. And I used a special type of cells named Muse. Muse is a sort of multilineage differentiating stress-enduring cells. Those cells exist naturally in our body, in dormant state, and they are activated when we experience significant stress or inflammation or injury. And then once they are activated, they will initiate regeneration cascade responses. So using a mouse model of gastrointestinal syndrome, I showed that when Muse cells are injected to mice after exposition to radiation, they promoted the intestinal recovery and survival of the animal, contrary to those that are not treated, indicating that Muse cells may be used as effective strategy to manage gastrointestinal syndrome after exposition to high dose radiation. Thank you.
And now, from the Lewis Lab in molecular pharmacology, Michele De Franco will talk about Copernic's precision strikes on tumor microenvironment.
What if I told you that fighting cancer doesn't always mean going after cancer cells themselves? Tumors build a fortress around them. This fortress is the tumor microenvironment, and it helps the tumor in its growth, invasion, and protects it from our best weapons. One of the chief architects of this very complex and dynamic system of walls are the cancer-associated fibroblasts. These cells all show on the same flag, called Fab, fibroblast activation protein. Fab is found to be highly expressed in over the 90% of solid tumors, but almost never in healthy tissues. And this is quite rare in oncology, isn't it? So, what is our strategy? Instead of attacking the castle core, why don't we target the walls? Why don't we target the tumor microenvironment? To this aim, we develop small, precise molecules, represented in this story as knights, able to target Fab with high selectivity and affinity. Then we armed our knights with a radioactive weapon called copper. We gave them at first copper-64 to better see the tumor using high-resolution PET-CT imaging. Because remember that we can fight what we can see. Then we moved to copper-67 to treat the tumor by delivering radiation right where it's needed. Doing this, we have one molecule, two different isotopes. both diagnosis and therapy in one. And this sounds to me like a true theranostic tool, right? And this is exactly what I've been looking at. Testing these molecules in multiple tumor models to see how well they can visualize and treat the tumor. And we got very promising results in both imaging and therapy, and all without harming healthy tissues. And this is quite remarkable. But, beyond the data, it's about the strategy. As I told you at the beginning of this story, tumors build fortresses around them. And now, thanks to our brave knights, we have a way to turn the tumor-owned defenses into a weakness, paving the way to new, more selective, effective, and personalized anti-cancer strategies. Thank you.
Our next speaker, Swati Jain from the Morris Lab in the Department of Surgery, will tell us how tumors got the balls, but we got the brains.
Ladies and gentlemen, welcome to the epic showdown between brains, brawn, and biology. In the center, flexing way too much confidence, is Mr. Tumor. He's bold, he's brash, and he's got the balls. But don't be fooled by that big cheesy smile. Mr. Tumor has a secret. He's not just any lump of trouble. He's got something very unique, something that's usually found downstairs. If you catch my drift, I'm talking about cancer testis antigens, or CTAs. That's right, these antigens are normally found only in the testis. But thanks to an out-of-control genetic program, tumors start making these testis-only proteins and flaunt them like some fashion accessories. But here's the catch. These same fancy proteins make Mr. Tumor stand out like a guy wearing a tuxedo to a summer rooftop party in New York City. Totally out of place, right? And that is what makes them perfect cancer targets. Some of the CTAs even help Mr. Tumor grow bigger and stronger. But in the imaginary world where Mr. Tumor thinks that he's invincible, enters the scientist. She's sharp, she's focused, and she's already a little crazy from her PhD. So, no time for nonsense. With just MSK's mission, ending cancer for life, hardwired into her brain, she has been crafting a plan to train our immune system to hunt down Mr. Tumor's fancy proteins with precision immunotherapy. And she's got fancy stuff too in her toolbox, like engineered CAR T cells, antibodies. She can even design cancer vaccines to target CTAs. And don't worry, guys, CTA-targeted therapies do not harm the testis. Why? Because testis is naturally an immune-privileged organ, just like the cornea of your eye or your brain, shielded from immune attack. And what makes this approach even cooler is that CTAs pop up in a wide variety of cancers. That means they have the potential to inspire universal cancer therapies to treat all kinds of cancers. So yes, tumors may have the balls, but patients have the guts to fight the cancer. And we scientists have got the brain to turn a weird biological mistake into a brilliant cancer fighting strategy. Thank you.
Our next speaker, Erin Santos from the Broach Lab in the Department of Surgical Quality and Safety, tell us, Bon Jovi asks, who says you can't go home?
At Memorial Sloan Kettering, most surgical patients have a predictable post-surgical recovery course, averaging about 6 hospital length of stay days in 2023. However, there were 235 patients who stayed longer than that, 10 days, more than 10 days in the hospital, accounting for 1,700 hospital days. This places an enormous burden on the hospital resources. Most of these patients undergo complex surgeries, such as debulking or removal of tumor lesions from multiple organs in the body, such as the bowel, the spleen, or the diaphragm, or pelvic exenteration, or the removal of multiple organs in the pelvic cavity, like the bladder, the rectum, uterus, and ovaries. These surgeries have a high risk for complication. So while 81% of these patients with a prolonged length of stay because of complication management, 19% of them that year stayed because of pending coordination of care needs. To address this, we established a complex surgical recovery team. This is a multidisciplinary team with colleagues from these surgical departments and non-surgical departments coming together to provide support for providers and patients with complex and prolonged post-surgical recovery. After 6 months of piloting this with gynecological surgery service, where we had great buy-in, we've enrolled 9 patients, totaling 54 recommendations and averaging 6 recommendations per patient. Most of these recommendations are on nutrition, so making sure the patients are on early oral diet to facilitate bowel motility and faster recovery. Second is early discharge planning, making sure all the services and supplies that they need to recover at home are in place and determined at the early part of the admission. And third is wound care. Do the patients have the best wound care regimen that's evidence-based to facilitate their healing? I'm happy to report that we've decreased the extra length of stay from 30 days to 2 days. We plan to review our outcomes 1 year from our pilot and hope to expand this to other surgical services to continue our mission of improving the care for our cancer patients. Thank you very much.
And now, from the Choi Lab in Developmental Biology, Brendan Camellato will tell us how molecular recording revealing the hidden rules of cell fate.
We all begin life the same way. It's just a single cell. That cell divides and develops, eventually into trillions of cells with different functions, all working together to make us. And much like our lives are shaped by things like our environment, our interactions, and challenges that we face, throughout development, each cell experiences its own journey, interacting with its environment and with its neighbors, responding to signals, and adapting to all different kinds of stresses. But we still don't fully understand how all these different influences combine within each single cell to determine if it will become a brain cell, a liver cell, or a tumor cell. In the Choi Lab, I'm developing tools for molecular recording to give each cell the ability to record its own history. We can rewire a cell's natural responses to these different experiences so that when something important happens, it makes a small edit to its own DNA, leaving a lasting note. Each cell can write these notes continually over its lifetime, keeping a record of all the important things that happened to it. I can then sequence each cell's DNA, read out the series of notes that it left, and reconstruct the story of how each cell came to be. Here's an example. We have a stem cell that can become either a liver cell or a pancreatic cell, but we don't know exactly how it makes its choice. With molecular recording, the cells can record hidden steps along the way that we would otherwise miss just by looking at the final outcome. Say, for example, that a brief exposure to low oxygen pushes the stem cell into an intermediate state, then committing to become a pancreatic cell in response to a specific signal. The stem cell, responding to the same signal, but without going through the intermediate state, instead becomes a liver cell. These unique histories are written as distinct records in each cell's DNA, allowing us to read out the specific sequence of events leading the stem cell down one of the two paths. And this isn't just curiosity about human development. Understanding cell fate choices has real-world implications. If we know the exact sequence of events that produces healthy liver or pancreatic cells, for example, we can reliably recreate those conditions in the lab, producing healthy cells to repair diseased or damaged organs. With cancer, we could identify a critical tipping point when a healthy cell turns cancerous, maybe a specific moment after its DNA is damaged and it receives an inflammatory signal. This could reveal when and how to intervene before the disease takes hold. Confucius said, "Study the past if you would define the future." With molecular recording, we can do just that, revealing each cell's past to better understand development, catch disease, and shape the future of medicine. Thank you.
Now, from the Lewis Lab in Molecular Pharmacology, Shaniqua Lawson will tell us from clues to cures, multimodal imaging for ovarian cancer.
Every year in the US, nearly 21,000 women are diagnosed with ovarian cancer, and sadly, 13,000 will die from it. Ovarian cancer accounts for 2.5% of cancer cases in women, but 5% of female cancer deaths, having the highest mortality rate of cancers involving the female reproductive system. It's often caught late, when the odds are already stacked against us. Only about 20% of these cases are caught early, and here it's pretty easy to treat. Unfortunately, most patients are diagnosed at advanced stages, where the 5-year survival drops to 30% or less. Surgery is the main treatment, but tumors can hide. Missing even a few cells means the cancer could come back. And even when surgery removes visible tumors, 70% of patients still experience relapse or recurrence, especially in these advanced cases. And honestly, surgery hasn't changed much in the last 150 years. It still just boils down to steady hands, a sharp knife, and a good pair of eyes. My research is about giving these surgeons better eyes or better tools to see these tumors. So let's think of ovarian cancer like a mystery. Antibodies will act as our lead detective. They're very specific in their binding, so they already know what to look for, like overexpressed markers on ovarian cancer cells. So they're the ones going to the crime scene, gathering the evidence, and creating the case file. PET scans are like that first clue. They create a map of the crime scene where important information is highlighted in red, or they can show where tumors are hidden in the body. But once you're in the crime scene or inside the body during surgery, you need more information. That's where near-infrared fluorescence or NIRF imaging comes in. It's like shining a black light directly into the crime scene, allowing you to see what's hiding inside. During surgery, tumors are lit up in real time, and so the surgeon can clearly see tumor margins, almost like evidence in a crime scene glowing under a black light. Clear margins means the surgeon can remove more tumors much easily, reducing the chance of it coming back and improving patient outcomes. My research is about making our lead detective, the antibodies, smarter and better equipped. So instead of carrying 2 agents, one for PET and one for fluorescence, I'm developing multi-purpose antibodies that can carry both tools at once. So here, we have one molecule with both capabilities that we can easily add to the antibody. This way, our detective can go to the crime scene with the map in one hand and the black light in the other. This is vital in ovarian cancer, where disease can spread in tiny, hard-to-see patches throughout the abdomen. So with the PET, this allows us to scout out the area in advance and map out the tumors. And then the fluorescence allows us to look closer and light up the scene during surgery, so no clues are missed. Our lead detective, the antibodies, they make sure the evidence is real and specific to ovarian cancer cells. The result? Surgeons can remove more tumors in a single surgery, leading to fewer missed cells and better patient outcomes. So in short, I'm using antibody-based multimodal imaging in order to go from hidden tumors to visual clues, giving surgeons information in real time and making sure that patients can get the best chance at having a clean slate. Thank you.
Our next speaker comes from the Seton and Powell Labs in Department Radiation Oncology. Pooja Yadav will talk about control copy, control chaos.
Hi, I am so excited to be here. Oops, I'm really sorry for my duplications. I hope it wasn't too bad, because for cancers, they really are. In some of the most aggressive and hard to treat prostate and ovarian cancers, there's a strange pattern in their DNA. A large chunk of the genetic code copied and pasted next to themselves. These are called tandem duplication. This pattern is strongly associated with a gene called CDK12. When a cell loses both copies of CDK12, it makes these errors again and again. But why and how? That's the mystery I am trying to solve. We think tandem duplications come from DNA replication-associated damage. When a cell copies its DNA, sometimes the replication machinery stalls, something called replication stress. It may result in or be caused by small breaks or nicks in the DNA. In a healthy cell, these nicks are rapidly patched up. But in a cell where CDK12 is missing, we suspect this repair process goes wrong. To test this, we are using the modified version of the famous CRISPR-Cas9 tool. Unlike Cas9, which is used to cut both strands of DNA, this one makes nicking just one strand, mimicking the kind of damage that can naturally occur when DNA replication stalls. This lets us ask two main questions. First, do these replication-associated breaks actually cause tandem duplication? Second, which repair pathways or lack thereof are responsible for converting these small lesions into large-scale genomic chaos? By identifying key mechanisms and pathways involved, we can then develop specific targeted therapies, giving patients with CDK12 loss who are very aggressive cancer, very difficult to treat, a renewed hope for better treatment outcome. So yes, I did duplicate it a bit in the beginning, but unlike cancers, mine was deliberate, short-lived, and maybe a little helpful, maybe for my scores. Thank you.
Our next speaker comes from the Caleb Lareau Lab in Computational and Systems Biology. Arthur Shao is going to talk about constructing a Formula One champion CAR T-cell from scratch. Arthur.
CAR T-cell therapies have completely redefined what the possibilities are for cancer treatment. And MSK has been the pioneers of this, pioneering innovations in designs that have led to FDA-approved treatments for multiple types of blood cancers. But what exactly is a CAR T? Well, it stands for chimeric antigen receptor, and it's essentially a custom sensor that can reprogram a patient's own T cells to be able to recognize and attack cancer. But as powerful as CAR Ts are, they're far from perfect. In fact, many of the current approved therapies today still struggle with consistency, performance, and durability. But that's where my work comes in. You see, the challenge lies with one of the most critical components of the binder, the CAR T, known as the binder, which you can think of as the molecular grip that latches on to the cancer cell. Traditionally, these are just binders that are borrowed from old existing antibodies that were never designed for the task at hand. They can take months to develop, can be very unstable, and oftentimes can cause unwanted toxicities in patients. To put that into perspective, that's like entering a modern Grand Prix this year with last year's engine. You've put yourself at a huge disadvantage, and now you've given the opposition an early lead on you. So what's the solution? Well, I built a platform to be able to design and test binders as if I had my own Formula One engineering team. Instead of recycling old parts, I'll start fresh using AI-driven protein design tools to be able to build fully customizable binders completely from scratch. But design is just the first step. Just like a race car needs track laps, even the best designs need further fine-tuning. And that's where this rapid screening platform comes into play. I can test multiple designs in parallel at the same time, and I can be able to test them at high throughput. And I judge them based on these four key metrics. The first is affinity, which tells me how tightly the binder or the CAR T grips the cancer cell. Second is specificity. How well does it avoid any off targets? Third is tonic signaling. How functionally stable is the binder? And lastly, tumor killing. How well does it do the job where it matters the most? I then take all four of these components into a single composite score, where I can then evaluate and rank each binder to determine the best one. And the result is a race-ready CAR T right out of the gate, fully optimized, equipped with the features it needs to successfully beat cancer. In Formula One, the right engineering can turn a middle-of-the-pack race car into a championship contender. In cancer treatment, the right engineering could be the difference between a therapy that falls short and one that can actually make a difference in patients' lives. Thank you.
And now from the Kolesnick Lab in Molecular Pharmacology, Radhika Rao Arasala is going to tell us the curious case of methylation, anti-aging button.
Good afternoon, everyone. I'm sure most of you must have watched the movie The Curious Case of Benjamin Button. It's a fantastic movie. In this movie, the character Benjamin Button was born old and he aged backwards. That is, he becomes younger with time. It sounds pretty wild idea, right? What if I say I could do something similar in my research? Let me explain in a very interesting way. Imagine your cell as a big city, and there are these long, huge roads that leads to different destinations. The roads being your DNA, and the destinations are different cells and their unique functions in our body. But not all the roads are always open, because there are traffic lights at every intersection. What do these traffic lights do? They control the traffic flow. Similarly, in our cells, we have specialized proteins called histones. Like the traffic lights, they control the protein production. When there is a green light, the protein is produced, and when there is a red light, the protein is not produced, which we call as gene regulation. But how are these traffic lights flipped from red to green? Like the traffic system, in our cells, we have traffic controllers. So these traffic controllers, basically, they add these specialized chemical groups to the histone proteins or the traffic lights, which helps in switching to produce the protein or not to produce the protein, which makes each cell unique. While studying these traffic controllers, I discovered a novel function that not only they help in protein production regulation, but they also decide where the DNA has to be positioned inside the nucleus of a cell. And it became more fascinating when we identified that with human ages, that is from young to adult to old, the number of traffic controllers become fewer in number. As you can see here, an old cell, without the traffic controllers, the entire system has collapsed. There is traffic jam and the cell is not working efficiently anymore, which we call as cellular aging. So I asked the question, what if I add these traffic controllers back into an old cell? Can I reverse the aging like the character Benjamin Button? And guess what? Once these traffic controllers were added back to an old cell, the traffic was controlled again and the protein production was regulated. The DNA was pushed back to its original position. So we could say that these traffic controllers are acting as an anti-aging button that can push to work an old cell to behave younger again. Thank you so much.
With that, I want to ask our fellow judges to walk out of the room with me to deliver in the other room. And I want to pass the podium to my colleague, Dr. Ina Bakman-Sanchez, who's going to entertain you for a minute or two while we deliver all together outside of the room.
So, hi everyone. This is your opportunity now. I'm just going to remind you of all our speakers today. Get your phones out. I'm going to give you 2 minutes. This is the fun part, right? Usually asked to put them away. And as you're ready, you can either type the tiny URL or use the QR code. You can vote only once, so take a minute, decide who you like. And yes, we always encourage the participants to vote. Please do. So I'll give you 2 minutes. With that, I want to move to our second favorite initiative during the Postdoc Appreciation Week. Every year, we get to ask our postdocs to show their creative side, and specifically how they see the world. So through their eyes, we got a submission of 24 photos. And again, the community here had the hard time to choose the top 3 winners. I would like to thank our winners, and I realized that Sandy left with the certificates. Well, it happens, right? No event without hiccups, and thankfully those are the smallest ones we have. But if we do have our winners, yeah. I mean, I remember that day, not the grandma part, but being in the labor room and how everyone around me were going crazy, especially the grandmas becoming. But I do want to give a big applause and a shout out to Megan Kelley, who also presented today, but submitted our first place winner photo. And can I at least shake your hand? And penny for our thoughts. This was a really great capture. Our second place was from the Sfeir Lab, Zhangdong Kao. If you are here, you are here. All right, I'm not going to make you come down, but congratulations. May your generating stations for science never stop, and may it kiss the skyline with a touch of beauty. I thought the title was phenomenal for this. Third place, Manendra Lankadasari. If you're here, let's give you, here you go. Congratulations. Okay. Who needs Wi-Fi? Well, I can tell you, definitely me yesterday when mine crashed. But who needs Wi-Fi when the universe is streaming live above your tent? I was just curious, where did you take this photo? Oh, right. Beautiful. I was like, I haven't seen the sky like this, well, since I moved to New York. Now, I did want to give another shout out to 2 photos that were submitted that were very, very tight with our winners. So, Corey Haluska from the Zhao Lab, beautiful photo when the storm retires, the sky writes apologies in gold. And I'll give Corey a shout out. He won the previous 2 years. So keep submitting those beautiful photos. And Luan Lee from the Soldatov Lab bringing us the beautiful Brooklyn Bridge and telling you to keep your research and connect the dots just like the bridge is, because one day you'll be as famous and get to be featured on Hollywood. So, congrats. Now, you all know we try to bring some fun with our appreciation video, and this year we thought it would be a good moment to reflect on all the good times that we had throughout the years. So I want to bring you back the memory line and just enjoy.
postdoc. Mentor. Mentee. One person. Member. Lab member. It's getting small. A cell.
Cell interaction.
Cell therapy. Lasers. Yeah. Something having to do with, chemotherapy? No. Immunotherapy. Go team faculty. One word. You. Postdoc. Person.
Trainee.
Human.
Scientist.
Circle. Cell. The globe.
Liquid phase separation.
Okay. Coffee. Scientist. Lab.
Researcher.
Drinking coffee. Coffee break. What's in there? Water? Oh, vapor? Major spill. Minor spill. Skin, tissue, epidermis. A skin container? What?
Of water / coffee.
It was plasticity.
Good afternoon. My name is Richard Dawson, and I'd like to welcome you to Postdoc PI Family Feud. Thank you so much for joining us this afternoon.
You were asked was, on which day of the week should lab meetings be held? And your answer was? Survey says 28. From MSK leadership, who should be the next member of the Avengers? You went with the obvious answer, which was
Top choice.
Ushma. Survey says. What is the Greek letter used most frequently in science? I believe you went with a pandemic-themed answer of Omicron. Survey says. Well, you can imagine people might. Other than coffee, what is the drink that's most consumed by scientists? You did not go with Red Bull, but instead you went with beer. Survey says. And what is the best model organism? Let's see, E. coli. Let's see what people said. Survey says. So a grand total of 83, which is really impressive. Let's give Anna a big round of applause.
we do have the certificates for the photo contest winners. So can I get all three of you here quickly? And just to show those photos. So, Megan. Congrats again. Okay, I'm gonna give the podium to Jason to announce the winners.
All right, drum roll, so I don't actually know what's coming next. There are my glasses. Okay.
I'm gonna announce all the participants that didn't win a prize.
I'm going to announce all the participants. See, Yahara's in charge. I'm going to announce all the participants who didn't get a prize. Pooja.
Everyone.
And everybody come on up. Here, we get a big photograph of everybody. Honorine. Swati. Everybody gets a certificate this way. Shaniqua. Erin. Michele. And Radhika. I just want to say everybody was absolutely fabulous. I think that thank you to the judges. I heard you had a very, very tough time. So thank you all very much indeed. And same time next year? Yeah. Okay. All right. Brilliant. Okay.
The prize winner, just stay there.
Just stay there. Okay. Okay. So in third place, Arthur. In second place, Megan. And well, in first place, Brendan. The boys got one. And don't go anywhere, Brendan, because you also got the People's Choice as well. Wonderful. Thank you so much again. That was fantastic. And, oh, don't forget your check. There you go. Congratulations. You have to pay half in taxes, so don't spend the whole 3,000 yet. Don't deposit that, yeah. So again, thank you so much, the judges. I think this was yet again just shows the phenomenal standard of our postdocs at MSK. Congratulations to all of you for all your hard work and really just doing a great job today. And right now, outside, we still haven't had a baby yet, but you can ask Sandra how that's going. But right now, everybody's invited to outside for some drinks and some food. And thank you all for attending today. Thank you very much indeed.